All right, so after today, only two more sets of lectures to go. Since we finished Chapter 8, which is plasticity, I did seek feedback and ask, what you guys wanted to talk about with the rest of our classes. And I think what I'm going to do based on your feedback is to discuss different neurodegenerative diseases, okay? There are many to talk about. We'll see how far we can get. I'm going to start with Alzheimer's and then probably talk about Parkinson's. There's a lot of detail in the book about Parkinson's disease. And then we'll see if we can get to ALS, amyotrophic lateral sclerosis, as well as MS, multiple sclerosis. But again, we'll play that by ear. The book has varying degrees of detail. So like I said, there's a lot on Parkinson's. There's relatively little on Alzheimer's. So I might bring in other figures or things of that nature to try to augment what the book has. I'm going to start with Alzheimer's, and we're just going to start by talking about epidemiology. We'll talk maybe about the genetic basis, and then I'll introduce some cellular mechanisms. What is in the book is found on pages 713 and 714. So if you want to supplement the notes, you can take a look at what's in the book. Okay, so Alzheimer's disease, as you probably know, triggers a certain type of dementia. So again, I think you probably have some sense of what this word means, but when we refer to dementia, we're talking about something that's characterized by recent memory failure, as well as other loss of intellectual functions. So it's the memory failure that gets the due, right? That's sort of the characteristic, but there are other sorts of intellectual functions, cognitive functions that decline as well. And so the most common type of dementia is caused by Alzheimer's. Okay, it's the most common type. How common? Well, about 5 to 10% of those people over 65 develop Alzheimer's over the age of 65, and up to 45% or more over 85. Okay, so this is age onset. Interestingly, the only way we can truly diagnose Alzheimer's is through post-mortem brain tissue analysis. So we can only get a concrete diagnosis when we examine the tissue from someone who's deceased. Now, we can look at symptoms and say those are symptoms that are very consistent with Alzheimer's, so we can diagnose it based on symptoms, but we can only really truly give a concrete diagnosis after death or post-mortem by looking at the histology, that is the tissue that has degenerated. And so when that's done, there are really three things that are seen. One is the presence of these intraneuronal neurofibrillary tangles. So we see intra, that is within neurons, neurofibrillary. Tangles. These tangles are cytoskeletal fragments that bind together. So you can see them here in yellow. Right? So these are cytoskeletal in nature. So that's a characteristic of the histology. The second thing that's seen are protein aggregations of amyloid protein. So amyloid is a specific type of protein, and we'll talk about this. Protein aggregations. These are sometimes referred to as plaques, right? So there's a large intracellular plaque that forms. And then the third thing that's characteristic of a postmortem brain is a really diffuse loss of neurons. And that's what's shown in B, is all of the places where neurons are degenerating and dying. So in places like the hippocampus, which we know is important in memory formation, in the amygdala, which we think regulates emotional state, and all over the cortex, right? So this is the outer portion, right? So this is the cortex, and you can see selective loss in the cortex as well as around here, limbic system regulating emotion, hippocampus, broad effects of cell death. The majority of cases of Alzheimer's are sporadic. This means we don't exactly know why they occur. Additionally, they're late onset. So again, this is typically seen in older adults versus younger adults. However, there are some early onset cases that are monogenic, which is great. Well, not great, but really useful in the study of Alzheimer's. So some early onset cases are monogenic. You probably know what that word means, and if you don't, you can gather, right? Mono, one gene, so they're controlled by one gene. And this really allows for the study in animal models. Now, we might not be able to study the cause in these sporadic cases as easily, but we can study the effects by manipulating genes in mice or rats, right? Mutating the same genes that we know cause it in humans, and then looking at the effects down the road. The belief initially was that the genes that are related, even in late onset cases, are on chromosome 21. So the changes, mutations, maybe that's a better word. And the reason that this was believed to be true is because those who have neuropathic features of Alzheimer's, so those with the neuropathic features, right? Basically what they're displaying, the pathology, the disease state in the brain, these neuropathic features are similar to those with trisomy 21. You might have heard of trisomy 21, right? This is better known as Down syndrome. And so, due to abnormal meiosis and chromosomal non-disjunction, right, someone who has trisomy 21 literally has a third chromosome at that 21st position. Okay, so you know that chromosomes have homologs, right? So 21 and 22 are homologous, and we have 46 total. Well, there's actually 2 copies of chromosome 21. And again, the identifiable features from a cellular level look similar to some of the cellular changes in Alzheimer's disease. So in studying those with early onset Alzheimer's, we were able to get some understanding of the genes that we think are involved and the proteins that those genes code for. The first is that the gene encoding amyloid protein becomes the focus, right? This is called APP. So APP is amyloid precursor protein. Okay, and mutations in this gene, which is on the 21st chromosome, are linked to these early onset Alzheimer's cases. We know that there are amyloid deposits, right? These plaques, and here's one here, right? These plaques are characteristic. These are hallmarks of Alzheimer's disease. Furthermore, a portion of APP amyloid precursor protein, which is called A-beta, amyloid beta, right, is part of these plaques. So it's not the entire APP that is the plaque, but you can imagine that some part of it is cleaved, and that cleavage product, A-beta, is what's found in the plaques. Other genes identified in autosomal dominant Alzheimer's disorders were presenilin 1 and presenilin 2. And I should stop there because I mentioned it and I didn't mention it before, that these early onset cases, right, they're autosomal dominant. Right, and if you remember basic genetics, right, and if we go down here and we're going to say someone who's a heterozygote for the amyloid precursor protein gene, right, the mutation, this person would be afflicted with the disease, right, because it's autosomal dominant. You don't need two recessive alleles to impart the disease. All it takes is one dominant allele. Okay, so even a heterozygote would be afflicted with this. Okay. Okay, so jumping back to presenilin 1 and presenilin 2, these genes, and I'll talk more about them, code for proteins that modify or process APP protein. So the proteins, the protein products of the genes, APP cleavage products. Okay, so exactly how APP gets cleaved is going to be somewhat dependent on presenilin 1 and presenilin 2. And so we get an increase in A-beta and a particularly toxic form of A-beta, a peptide called A-beta 42. Okay, so similar to A-beta, but this is referring to the number of amino acids in the peptide. So basically, and I'll show you an example of this, or we'll go through this in a minute, but you have the APP protein, and depending on where it gets cut, right, certain portions of that may form A-beta or A-beta 42, and these are what tend to form or aggregate in plaques. Okay. I think this information is all well and good with respect to APP and presenilin 1 and 2, but the problem is, right, remember, this is only looking at early onset cases of Alzheimer's, and we want to know what the genetic basis is potentially of those folks who have late onset, and so that's what we'll talk about next.